Our article was Carbohydrate and Fat Intake Associated with Risk of Metabolic Diseases through Epigenetics of CPT1A, presented by Athena Breo, Sydney Kaler, Marissa Mall, and Tori Simmons.
The hypothesis of this study is that total dietary carbohydrate and fat intake affect methylation of CPT1A, leading to altered CPT1A expression and altered risk of metabolic diseases like obesity, type 2 diabetes, hypertriglyceridemia, hypertension, and then overall metabolic syndrome. So the gene of interest in this study is CPT1A, which is carnitine palmitoyl transferase 1A. It is the key enzyme in the carnitine-dependent transport across the mitochondrial inner matrix. So the CPT1A gene is an enzyme that converts long-chain acyl-CoAs into long-chain acylcarnitines, which allows them to be transferred from the outer mitochondrial membrane into the inner mitochondrial membrane and into the mitochondrial matrix for fatty acid beta oxidation to occur. When there's decreased activity of CPT1A, there is a decreased rate of fatty acid beta oxidation, and high levels of methylation at the CG00574958 region of CPT1A have been found to be associated with reduced risk of those metabolic disorders like obesity, type 2 diabetes, and abnormal triglyceride levels. So a little overview of the gene. It's protein coding. It has about 90,000 base pairs, 22 exons. It's located at chromosome 11, and it's expressed in several tissues. And aside from just fatty acid beta oxidation in and of itself, it can regulate functions such as insulin-mediated inhibition of glucose production, insulin secretion, glycogen synthesis, and appetite control. So previous association studies have identified DNA methylation of CPT1A and have found it to be associated with reduced risk of metabolic diseases. And early studies conducted in rodents found that carbohydrate intake in that study, which was high fructose levels, induced DNA methylation of CPT1A, while high fat intake suppressed DNA methylation of CPT1A.
So this study was a meta-analysis of 3 cohort studies, the Genetics of Lipid-Lowering Drugs and Diet Network, or GOLDN study, the Framingham Heart study, and the REGICOR study. So originally in the GOLDN study, they were trying to determine the correlation between carbohydrates and fat intake and methylation at CG00574958 in the CPT1A promoter. Then they replicated this in both the REGICOR study and the FH study. So they extracted DNA for methylation from whole peripheral blood from both the REGICOR study and the Framingham Heart study. And then in contrast, DNA was isolated from the CD4 plus T cells from frozen Buffy coat in the GOLDN study. So the GOLDN study was part of the Framingham study originally, and the main objective was to identify the genetic variants that accounts for an individual's lipid response to a high-fat meal and 3-week treatment with fenofibrate, which is a triglyceride-lowering drug. Originally, it was 1,327 men and women ages 18 and older, and then 978 of those participants gave peripheral blood samples collected for the genome DNA methylation data analysis. In the Framingham study, the main objective was to identify key factors that determine cardiovascular disease. It was originally 5,124 participants, and both the children and spouses of the original cohort was included in that. Out of that, 2,331 participants' peripheral blood samples were collected for genome DNA methylation for data analysis. And then finally, the REGICOR study, the main objective was to investigate investigate risk factors of cardiovascular disease, and 4,980 participants were in that original study, and then 645 of those participants were donated their blood for DNA methylation data analysis. So the primary outcomes of this study was metabolic diseases such as hypertriglyceridemia, obesity, metabolic syndrome, and type 2 diabetes. And then the secondary outcomes were hypertension. Analyses conducted were based on linear or logistic regression models, and then the software utilized was SAS 9.4 or SVS 8.7. And then missing data was omitted for all statistical analysis.
All right, now for the results of the study. First, we're going to look at the results by characteristics. So they looked at age, population, and dietary intake. With age, they found that the youngest population, which was the GOLDN study, was less likely to have metabolic disease, while the other two, the REG-ICOR and FH, were more likely to have these metabolic diseases. When they looked at population, the GOLDN and FHS population was from the US, while the REG-ICOR was a Spanish population, and they found this to be correlated with dietary intake. The Spanish population was more likely to consume more fat in their total diet, while the other two were more likely to have total carbohydrates be higher. Now looking at the CG00574958 methylation at CPT1A, they found a negative association with all of the metabolic diseases. And then on the next slide, we'll see kind of the results in a chart. So the variation in the gene was positively and significantly associated with carbohydrate intake normalized to the total energy intake, all with significant p-values. And then the methylation also was positively correlated with the ratio of carbohydrate and fat. Looking at the total carbohydrates on the top line, you can see that the intake remained the strongest association with the CPT1A CG00574958 methylation. And then also total fat exhibited the strongest association with the methylation. And this was the same when it was compared on an individual population or the meta-analysis in all three populations. Continuing the results, the carbohydrates were negatively associated with the CPT1A expression with a significant p-value of 0.0007, whereas the fat intake was marginally and positively associated with the CPT1A expression, and that p-value was 0.0169. The ratio of carbohydrate and fat was negatively associated with the CPT1A expression. And moving on to more results. So table 3 is basically showing us that carbohydrate intake showed a negative direct effect on all of these metabolic outcomes, which again were glucose, BMI, type 2 diabetes, but not hypertension. As you can see here, the negative effects were not shown with hypertension, and the MET-S. Carbohydrate intake had a positive direct effect on triglycerides in both the GOLDN and FSH studies. And then also in both of those studies, carbohydrate intake had a significant and negative indirect effect on all metabolic traits that were examined, which again were triglycerides, glucose, BMI, type 2 diabetes, hypertension, and MET-S with p-values all significant. And this really just suggests that carbohydrate intake decreases the risk of metabolic diseases mediated through the CPT1A CG00574958 methylation. And then lastly, this methylation was correlated with fat intake that showed a consistent and positive effect on all of the metabolic traits. And they all had significant p-values. And this observation indicates that fat intake increases the risk of metabolic diseases mediated through the methylation of the gene.
So in conclusion, this study demonstrated that carbohydrate and fat intake both have a strong association with CPT1A CG00574958 methylation and CPT1A expression, and overall the intake of both of these influences in some way the risk of metabolic diseases, more specifically through metabolic homeostasis. So carbohydrate intake, as we saw in the results, induces CPT1A methylation, and this leads to a lower risk for metabolic diseases and a decreased risk of insulin resistance. Furthermore, fat intake inhibits CPT1A methylation, which on the opposite hand increases risk for metabolic diseases. So overall, a balanced CPT1A activity is seen to be correlated with metabolic outcomes, whatever those may be. Some strengths of this study were that the initial analysis from the Golden study was replicated in 2 additional cohorts. And then there's a rather large sample size. It was about 4,000 subjects. Some limitations were that the mediation analysis was based on observational studies, and that the animal studies did show strong evidence that the high sucrose or high carbohydrate diets induce methylation at promoters of CPT1A, but future human studies are still needed. So the findings of this study should be just interpreted with caution. And lastly, CPT1A CG00574958 methylation and gene expression were based on the PBMCs, which are the peripheral blood mononuclear cells, rather than the most physiologically relevant tissue, which in this case was the liver. So finally, for future research, like I mentioned before, human intervention studies in the future are necessary in order to confirm the causal effects of high carbohydrate or high sucrose diets inducing methylation at promoters of CPT1A. Since energy metabolism is regulated by many pathways, carbohydrates could affect the risk of metabolic diseases through mechanisms other than the CPT1A enzyme. And this is possible since it was seen that the carbohydrate intake had a positive effect on triglycerides, but negative effects on metabolic traits and diseases. And then lastly, future research studies are needed in order to determine the actual exact mechanisms by which carbohydrate and fat actually modify CPT1A CG00574985 methylation. And these are our references. Thank you for watching.